Hello again. Let's take some time to talk about ATP. I know this is maybe more than one time that you've heard about ATP, and you're probably going to hear about it numerous other times. What I'd like to focus on in this particular discussion is the different ways that a cell might make ATP. So we're going to be talking about types of phosphorylation. First of all, remember what we said ATP is. ATP is a modified nucleotide. It's modified that it is in a triphosphate form. We can think of the bonds holding those terminal phosphate groups, especially the very last one, as being a special type of bond that might be referred to as a high energy bond. If I try to form an ATP, think about the energy required to push that phosphate group on. If we take a look at an ATP molecule, notice that in the representation of ATP, the bonds that are shown here are shown as a squiggly line to represent this special form of high energy bond. I almost like to think of them as being like a spring. A spring, for example, in some kind of toy that you would load by pushing or depressing a spring and putting a latch on it, catching it, maybe in a jack-in-the-box or in a dart gun, something like that. Now, those phosphate groups with all those oxygens, they don't want to be attached to each other. So I have to use a lot of energy if I'm a cell to force those phosphate groups together. Once together, that bond can be easily disrupted. Even though it takes a lot of energy to make it, it takes relatively little energy to get it to break. So as a result, I'm going to find that this terminal phosphate group in particular is going to be able to be used to store energy. Energy used to build the bond can then be released as the phosphate group breaks away from the ATP and moves to some other molecule, providing energy to that other molecule. Remember what we said about ATP molecules. We said that they're a way to store energy short term. Here's a triphosphate, adenosine triphosphate molecule with its phosphate groups. If it experiences hydrolysis and we remove that terminal phosphate group, there's going to be energy released. This process of turning ATP into ADP, remember we said, is an exergonic reaction. Okay? That means that if I wanted to reverse this process, build an ATP, I would have to add energy into the ADP in the phosphate group to force them together. And that's one of the things we want to consider, we want to focus on in future discussions is, how does the cell make these ATPs? Where does the energy come from to force a phosphate group to attach to an ADP? That's what we're going to be interested in. As a matter of fact, the way we classify the different types of phosphorylation events is by where the energy comes from to build that bond between a phosphate group and ADP. Okay? So, it's a key short-term energy storage molecule. You could also think of it as an energy carrier molecule or a go-between. Remember we said that ATP was going to be instrumental in forming that connection between coupling exergonic reactions that occur in one part of the cell, releasing energy to make ATPs, and then the ATPs breaking down to provide energy to endergonic reactions at some other location within the cell. So remember, we said that ATP was this energy go-between, an energy delivery truck, a coupling agent that allows energy to be moved from one region of the cell to another region of the cell. And, of course, we're going to be looking for how that happens in the cell. We call the process of building an ATP phosphorylation. All right? And so, well, first of all, let me say this. Phosphorylation in general means adding a phosphate group to something. So as you study other topics and other aspects of biology, you're going to find phosphorylation talked about in that general sense. In our discussion right now, we're going to focus on adding phosphate groups to ADP. We're going to be looking at that specific type of phosphorylation event. Okay? More general idea, we're focusing on the production of ATPs, phosphorylation of ADP. So, what we're saying is that there's going to be different ways in which a cell might make ATPs. And we're going to find that there are going to be 3 different ways in which we're going to see cells making ATPs. What's called oxidative phosphorylation, right? An oxidation process is going to provide the power. Substrate level phosphorylation, a substrate molecule working with a particular enzyme, it's going to provide the power. And photophosphorylation, and as that name implies, light energy is going to provide the power to make an ATP molecule. All right, now, there'll be separate discussions of each of these kinds of events in future topics, but we want to look at them in a general sense and get sort of an overview for this process. Substrate level phosphorylation. Here what we're talking about is a substrate molecule. Remember, a substrate molecule is any molecule that works with an enzyme. And we're going to see in these processes that we talk about that every step in the process has an enzyme involved. That means that any molecule in the process could be referred to as a substrate. Well, let's say I have a substrate molecule that has a phosphate group as part of it. If that phosphate group can be transferred to an ADP, then I will have made an ATP, and the power, the energy, the phosphate group to make that ATP has come from a substrate molecule. And when we get ready to talk about the processes of glycolysis and the citric acid cycle or Krebs cycle, we're going to see substrate level phosphorylation occurring during a couple of the steps of those processes. So I'll point that out to you when we cover glycolysis. I'll point that out to you when we cover the citric acid cycle. But we're going to want to look for those events where a substrate molecule provides the phosphate group and the power to make an ATP. All right? Notice we're defining the process based upon who provided the energy to make the ATP molecule, a substrate in this case. We're going to have also a process called oxidative phosphorylation. Here what we're going to be doing is we're going to be stripping electrons away from molecules. They might be organic molecules, and that's what we'll focus on, things like NADH, FADH, and so forth. Or I'm going to be stripping electrons from inorganic molecules. And you see this in particular in some of the kinds of oxidative phosphorylation that bacteria do. They can take electrons from more than just organic molecules. We have to take them from organic molecules. Those electrons that we pull off of those molecules we could think of as high energy or excited electrons. Those electrons get passed through a series of proteins. These proteins act as pumps. They're transmembrane active transport proteins. And they're going to use the energy from those electrons to create a hydrogen ion gradient. Remember what a gradient is. More of something on one side of a membrane than on the other side of a membrane. So we're going to actually see the cell create this disequilibrium condition. More hydrogen ions on one side of a membrane, less on the other. That's going to be tied to the use of an enzyme called Atp Synthase, or more commonly today, Atp Synthase. Well, think about what the name is saying to you. This must be an enzyme that can synthesize, build ATPs. And we're going to see that that proton hydrogen ion gradient is going to be used to provide the power for ATP synthase to build ATPs. All right, let me recap this very quickly so you can see what's going on. Energy from electrons pulled off of molecules, oxidation, provides the power for a series of protein pumps. They create a gradient. The gradient then is used to power an enzyme's production of ATPs. But notice what started it all off. It was that oxidation event where we got the original electrons to power this whole process. That's why the production of these ATPs is called oxidative phosphorylation. Much more detail on this process, as you can imagine. You can read more about it in the book, but I'm going to have a couple different presentations that will deal with aspects of this process. Now, what we're going to find is that later we'll talk about this as being a component of an event called respiration. And we're going to find that at the end of this chain of proteins, there has to be somebody to accept the electrons that have been passed down the chain. So later we'll talk about whether or not the process is aerobic, whether oxygen catches these electrons at the end of this passage, or anaerobic, when I have some other substance catching the electrons, usually an inorganic substance, but something other than oxygen. And so we'll talk about that as part of anaerobic respiration. So you can see that this event of oxidative phosphorylation is going to be a component of, it's going to be tied to the events that we're later going to talk about as respiration, the chemical process that makes the ATPs. The generalized process is called chemiosmosis, and we'll talk about that in more detail in the future. Think of this today as me just introducing some of these concepts to you, all of which we'll deal with in some more detail. Well, let's consider the last type of phosphorylation, photophosphorylation. Well, interestingly enough, photophosphorylation also is a type of chemiosmosis. So we're going to see some similarities between photophosphorylation and oxidative phosphorylation, but there's going to be some important distinctions as well. As the name implies, light energy provides the power to make the ATPs. We're not saying that light energy gets turned into ATPs. Instead, we're going to see something very interesting happen and something very important. All life on this surface of this planet is dependent upon this event taking place. It's the key energy conversion process of life. It's what plants and algae and some bacteria do to convert light energy into usable chemical energy. Very important. We'll talk about some of the details of it later. Let's look at it in overview. I'm going to use light energy to excite electrons. As the electrons absorb light energy, they get excited, and then they're going to jump to an electron transport chain, similar to the one we saw in oxidative phosphorylation. The electrons are going to be used, their energy, to create a hydrogen ion gradient. The hydrogen ion gradient is going to be used to power ATP synthase to make ATPs. The ATP production process overall has been powered by light energy. But you can see that it has as a component of it that passage of electrons, the pumping of hydrogen ions, and then the hydrogen ion gradient being used to power ATP synthase. So in that sense, it also is a type of chemiosmosis. We call it photophosphorylation because the energy needed to start, drive, cause the process to take place is light energy. We make the ATPs as a result of that original power, light energy, photophosphorylation. I've been mentioning this idea of chemiosmosis, so let me just mention it one more time. And you think about finding it, looking for illustrations of it, paying attention to the presentations I'll do in future lectures on this key idea of energy production. Now, what are the components? There has to be an electron transport chain of some type. Groups of transport proteins embedded in a membrane that can push hydrogen ions across the membrane. How do they do it? They do it with the energy provided by electrons that are passed along the molecules of that chain. So, electrons moving down a chain of proteins is used to power the active transport of hydrogens across a membrane. Think of that as the first half of chemiosmosis. Electron transport chain creates a hydrogen ion gradient. Now, what can I do with that gradient? Well, we're going to see that gradient utilized to power the ATP production of an enzyme called ATP synthase. So once I've created a hydrogen ion gradient, that gradient can then be used to provide the energy for the production of ATPs. We're going to actually think of those hydrogen ions. We could also refer to them as protons. Go back and look at some previous illustrations and remind yourself of what a hydrogen atom is. So if I have hydrogen ions or protons, we can think of the energy they provide to ATP synthase as a proton motive force. Those protons move by diffusion and provide the power to the enzyme to create ATP. Now, as you can imagine, we're going to want to look at this in more detail in future lectures. So, let's consider a quick summary of what we've been looking at. It's mainly the production of ATP. We talked about it as being a modified nucleotide, a triphosphate nucleotide. We said that it's a key coupling agent that links exergonic and endergonic reactions within the cell. We said that one type of phosphorylation, the adding of a phosphate group to a molecule, is what we were going to focus on. And we were going to focus on, in particular, adding a phosphate group to ADP, making ATPs. We said that there were three basic ways you could do this, and they differed in terms of what the energy source was to make the ATP. It could be a substrate molecule providing the energy, substrate level phosphorylation. It could be the oxidation of an electron carrier molecule in oxidative phosphorylation. And it could be light energy itself, providing the energy for photophosphorylation. Notice, defined by what provided the energy to make the ATP. We also pointed out how both oxidative phosphorylation and photophosphorylation have as a central element this event called chemiosmosis, an event where a gradient is created. The gradient is used to power an enzyme's production of ATP. Look forward to talking about most of these topics in much greater detail in future lectures. Thank you very much.